So today we're going to talk about what's referred to as RNA splicing. And this is one of the three modifications that need to take place to the primary transcript in order for it to be competent to be a messenger RNA. Of course, we looked at the other ones. There was the cap, 5' cap needs to be added, and then the 3' poly A tail needs to be added, and then finally splicing. And the splicing is the removal of the introns and the joining of the exons. So if you look at below, this is actually a gene. So it's representing the DNA, so it's double-stranded DNA. You see the promoter there, that's the promoter for the gene, and then you have the transcribe region, which is in the red and the orange. And for some reason, these things are going to make messenger RNAs to encode proteins. In order to code proteins, you have to have the codons lined up one after the next for each amino acid to be linked together. And for some odd reason, the coding sequences are split up on the gene. They're not linked together. And the region of the gene that contains the coding information, the proper exons, the proper codons, those are the exons, the coding sequences, and then the non-coding sequences, they don't encode anything. We'll see it gets a little bit more complicated than that because sometimes they do. Those are referred to as the introns, and eventually those need to be removed. So this is kind of an illustration, again, of what I just said. So you have the 5' cap, you have the poly A tail, and what this is showing is a primary transcript with the exons. And there's four exons in here, and then you have the introns in between, which just represents the line. And those introns need to be removed, and the exons need to be joined. And the arrow that's going through that messenger RNA represents the unified coding sequence for the protein. Now, what I want to kind of emphasize is that this process needs to be incredibly accurate. We're looking at an RNA molecule, and so somehow the apparatus that's going to carry out the splicing needs to be able to distinguish between an exon and an intron, and as far as we're concerned, it's just RNA sequences, just nucleotides. So somewhere in there needs to be the signal to splice properly. And it has to be, as I mentioned, really, really accurate. Why is that? Because if you don't make the cut as accurate as you can, or accurate as it ought to be, then you may leave out some nucleotides, let's say from exon 2, or leave some extra nucleotides in from the intron. And the minute you do that, you completely destroy the function of the messenger RNA. So it needs to be really, really, really well-tuned and exact. Now, this happens on 95% of all messenger RNAs, all RNA polymerase II transcribed genes. And some of them have a few introns like this, or 3 or 4 exons like this, but some have many more. And we'll take a look at that in the next table. So here I show in this table just kind of the range that you see of genes, primary transcripts, and the number of exons that could be observed. So like I said, 95% of all RNA polymerase transcribed genes have introns, and if you look at the average, an average gene maybe has 4 exons that need to be brought together, but the range is quite phenomenal. So here I have a table where on the left-hand side it's the gene, we're just looking at 4. Then you have the size of the primary transcript in nucleotides, the number of introns that need to be removed, and then the final size of the messenger RNA. So if you look at insulin, for example, it's rather small. the primary transcript 1700 nucleotides. 2 introns are removed, and 400 nucleotides for the primary transcript. If you look at catalase, it's a little bit bigger, 3,400 nucleotides, 12 introns need to be removed, and it's 1,600 nucleotide primary transcript. We move on to factor VIII, which is a protein which is involved in blood clotting, and we see it gets much bigger and much more dramatic. 186,000 nucleotide primary transcript, 25 introns need to be removed, and the final size of the messenger RNA is 900 nucleotides. So we've ended up basically just making an incredibly long transcript, 186,000, only to very quickly destroy 175,000 nucleotides, leaving only 9,000. Now, if we look at dystrophin, which is, I think it's the largest primary transcript that's been identified as greater than 2 million nucleotides. An enormous amount of time and energy required to synthesize an RNA of that length. There's some estimates that it takes a cell around 10 to 11 hours to transcribe that much RNA. Greater than 50 introns need to be removed, and so think of the sophistication of dealing with all these exons that need to be ligated or put together, and without removing or adding an extra nucleotide, because that would completely destroy the function of the message. You would destroy a codon, destroy the message. And then finally, the size of the messenger RNA is 17,000 nucleotides, which is still pretty long. But what happens here is, after spending all this time and energy making this 2 million nucleotide transcript, we very quickly cut out and destroyed 1,983,000 nucleotides. So it seems like an enormous waste of material, of energy. So there has to be some, reasonable explanation or reasonable advantage as to why the cell does this. It also, you see how big these genes are, and that also contributes to the size of the genome. That is, some genes are quite large, and so the genome is extremely large. And there's one question as to why the genome is so large when we only are making 30,000 proteins, when it's really only 2% of the DNA. Well, some of the genes are quite large, so it takes up more space than we would have imagined. So what we want to look at is what signals the proper cutting and joining of these required sequences. Remember, it's just RNA. How are the introns recognized? How are they removed? So it turns out that there are specific sequences on the RNA that represent signals that signal the machinery to do the recognition and the cutting. So in terms of the RNA splicing, there are 3 central pieces or 3 central components that I want to focus on. The first is, in fact, there are specific nucleotide sequences on the RNA that signal where the exons and introns are. We'll look at that in a second. The second is what's called the spliceosome, which is a very large multi-protein complex, also contains RNA. So it's referred to as a ribonucleoprotein complex that participates in directing how these things are removed. And then finally, self-hydrolysis plays an important role. We know that the RNA is reactive and it can cut itself. And so what happens is, in many cases, the proteins that are attached to the RNA after the proper recognition, they position the RNA in such a way that it undergoes a self-cleavage reaction through the 2' hydroxyl. So let's take a look, first of all, at the sequences. So the question was, how on earth does the cell recognize what's an exon and what's an intron? Because there's really no difference between them. It just is RNA sequences. But it turns out that there are specific nucleotide sequences that are conserved between every exon and intron. And that's what's shown on this figure here. So the exons are shown in blue, the introns are shown in yellow. And so we have labeled exon 1 and exon 2. And of course, you can have 10, 20, 30, 50, hundreds of exons that need to be removed. But the finding is that at every exon intron boundary, there are conserved RNA sequences. So if we look at the left-hand side, exon 1, we see there's a sequence AG in the exon, and then G, U, uracil. R stands for, R is an abbreviation, stands for either A or G, AGU. That's a highly conserved sequence at the border of every exon and intron boundary at the 5' end. So we refer to it as this side on the left-hand side is the 5' splice site. The 3' splice site, which occurs at the 3' end of intron 1 and exon 2, there's also conserved sequence, G in the exon, and then in the intron, there's this whole series of what are called Ys. Y is either C or U. N is any nucleotide, so it's not highly specific there, but there is some pattern, but there's always a CAG. And then somewhere towards the middle of the intron, it's usually 50 nucleotides upstream from the 3' end. There is another conserved sequence, which is Y, again, C or U, U, R, which is A or G, and A and C. The A that's in red is really the most essential. And so the A is going to turn out to play a role in the splicing process. So the answer to how does the cell differentiate between exons and introns when they're just simply what they look like to us, random RNA sequences, is that there are specific signals or specific RNA sequences that illustrate where they are or point out where these exon-intron boundaries are. Now, these conserved RNA sequences are what are recognized by the spliceosome. And that's what I'm going to talk about next. They're recognized by the spliceosome. Actually, what they're recognized by are RNA molecules that are within the spliceosome that base pair with these conserved sequences. Now, before I move on, I just want to point something out. When we talked early on about the genetic code, and we talked about the function of DNA and the information contained in the DNA, what I said was that there were many, many levels of information encoded in the DNA. And the one that we think most about and the one that we know most about is the genetic code for protein synthesis. That's all worked out. We know the codons, we know the amino acids. But here what I want you to see is that there is additional information encoded in the DNA. And that is the location of the exons and the introns. This is nothing that we can just look at and easily say, oh, there's some information there. But that is information encoded in the DNA that once in the RNA dictates where the splicing is going to take place. Okay, so let's look at the spliceosome. So the spliceosome. The spliceosome is a very, very large multi-protein complex. It's actually made up of 5 individual distinct clusters of proteins in RNA. Each one of those distinct clusters is referred to as a ribonucleoprotein complex because it contains RNA and protein, ribonucleoprotein complex. Each one has 10 to 15 different proteins, and eventually they all come together. So we're talking about, well over 50 to 75 proteins in a single spliceosome, and each one contains a small RNA, which is referred to as a small nuclear RNA of around 150 nucleotides. So there's 5 different RNAs. Each one has a different sequence, and they have different names. So there's small nuclear RNA U1, U2, U4, U5, and U6. I don't know why there's not a U3, but there's not. Each one has around 150 nucleotides. Each one has a different sequence, and each one is bound by a different cluster of 10 to 15 different proteins to make these small ribonucleoprotein complexes. And then these 5 complexes come together to form the intact functional spliceosome. Now, the different ribonucleoprotein complexes are named after the small nuclear RNA. So there's ribonucleoprotein complex U1, ribonucleoprotein complex U2, U4, and so on. Now, the way these things work is that some of these small nuclear RNAs recognize the base pairs at the exon-intron boundaries and base pair with them. So the recognition of these sequences is through base pairing by these small nuclear RNAs. They're in the complex with these proteins, and they organize into the spliceosome. So what I'm showing you down below is a very simplified version, but just to kind of get us started, is that the border between exon 1 and the intron is bound by the ribonucleoprotein complex U1, which is base pairing due to base pairing with the U1 RNA. That's how it gets located at that precise position. The border between the end of the intron, the 3' end of the intron, and the 5' end of exon 2 is bound by ribonucleoprotein complex U2 by virtue of the RNA base pairing with that sequence. And then U2 also appears to bind in the intron where that important A was. There's a series of sequences that are conserved, but the A is the most important. I have that circled with red, and that is also bound by U2. But what you have to realize is that all of these proteins are binding on this and positioning this in such a way that the intron gets looped out and the exons are brought together. This is also illustrated in the in the diagram on the right, the color diagram that I took from the book. And what it shows again is the exon 1 is in blue, the introns are in yellow. There's that A, which is important in this whole process, and the intron-exon boundary at exon 2, bound by the ribonucleoprotein complex U1 and U2 by virtue of the base pairing, which is shown. Then it shows U6 coming off and U6 going on and U1 coming off. But the bottom line is that these things are positioned in such a way that the adenine that's in the intron, the reactive 2' hydroxyl of that adenine, is positioned in such a way, in such close proximity to the exon 1 intron boundary, it cleaves, that 2' hydroxyl cleaves the phosphodiester bond between that last G in the exon and the first nucleotide of the intron, and makes a new phosphodiester bond between the 5' end of the intron and that adenine at the 2' hydroxyl. It makes what's called a lariat structure, and I'll show you more about that later. So a cleavage has taken place. The exon has been cut off. It's actually cut off by the RNA itself, the adenine. And then other activities in the spliceosome then now cut the exon 2 from the end of the intron, the 3' end of the intron, and they bring those two exons together to make a new junction between exon 1 and exon 2. In the next slide, I'm going to show you my drawing of this, which I think you'll see it's my bias is kind of shows it a little bit more clearly than this one. That's why I like my drawings better than the book, but okay. So here's my rendition of this, which I think may help a little bit in describing what happened. So of course, these are some of the events that take place in the spliceosome. And it includes, as I've written, self-hydrolysis. And that's carried out by that adenine in the intron. And so what happens is, of course, this is all bound up in the spliceosome, but the intron is looped out, and the 2' hydroxyl of that adenine that's in the intron cuts between the exon and the beginning of the intron, and creates a new phosphodiester bond between that 2' hydroxyl and the 5' phosphate of that G, which is the first G in the intron. So there's the creation of this strange 5' 2' linkage, which is the lariat. It's like a loop. And now you see the exon 1 is free. It's not free because it's all held within the spliceosome, so it's not floating anywhere. But then what happens is there's another cleavage reaction that cuts the intron off of exon 2. That lariat is released. You can see it's just floating there now. It's destroyed. And then you get the joining. Again, the spliceosome is carrying this out. You get the joining of exon 1 and exon 2. We call ligation. It brings those two together and seals them. And then, of course, if you have many, many more introns and introns, this is happening along the length of the primary transcript. So this is just a little bit more detailed structure of the lariat. I just want you to have a sense of what actually is going on here, because everything we've seen is kind of these drawings which really don't describe what is actually happening. So we have the lariat structure drawn, by this loop. So what actually is going on there? Okay, so let's look at the drawing. So we have exon 1. At the end of exon 1, there's a hydroxyl because the cut has already taken place. Then we have exon 2 at the other side, and then we have, it shows there's a little line that demarcates the separation between the exon and the intron, and then it says 50 nucleotides, and there's that adenine. Okay, there's that adenine that's important, about 50 nucleotides upstream. And what this is showing is that it links through normal phosphodiester bonds to the next nucleotide, which is U, and the one below, which I'm not showing. And just imagine these are all nucleotides, phosphodiester bonds, going around this whole intron. I'm not drawing them all, I'm just drawing a few. And at the very 5' end of the intron, there's that G. It's drawn upside down. There's that G, where the phosphodiester bond between the G and the 3' end of the exon has been cut by the 2' hydroxyl of that adenine, and now generates a new phosphodiester bond between the 2' hydroxyl and the 5' carbon of that first guanine in the RNA, and that is the lariat. And the adenine, that adenine there actually it's a strange thing. It has two phosphodiester bonds, one through the 3' hydroxyl to the nucleotide below it, and one through its 2' hydroxyl to that guanine, which now makes the lariat. So let me just say, this is, this gets extremely complicated. We're looking at, we just looked at 2 exons being joined together. But when we talk about a primary transcript with 20, 30, 40, 50 exons, I mean, you can imagine this is intense. And what I've shown you is actually an extremely simplified version of what happens. So what I wanted you to realize, you don't have to know this, you don't have to memorize any of these things here, but I just want you to see the sequence of events in a single splicing event is extremely complicated. So you have up at the top, which shows the exon, the intron, they call it that A, they call it the branch point, and then you have the second exon, and then it shows it going into this formation, moving to the left, and all these ribonuclear protein complexes are coming off and coming on, and there's all kinds of other factors, and there's cutting and splicing, and it's just, working out the conditions for this has been intense, and we still don't understand it. So I cut a little summary from a paper that I was reading, and I just want to read this to you because it just illustrates the complexity of this stuff. The major pathway. The splicing of pre-mRNA occurs within a large, very dynamic complex designated the spliceosome. The 50 to 60 S spliceosomes are estimated to be 40 to 60 nanometers in diameter, have molecular weights in the range of 3 to 5 million. Small nuclear RNAs, U1, U2, U4, U5, U6, are some of the best characterized components of the spliceosome. It's easy to characterize RNA. Proteins are harder and known to play key roles not only in spliceosome assembly but also in the two catalytic steps of the splicing reaction. Over 150 proteins have been detected in spliceosomes, and only a subset of these has been characterized. So we don't know a lot about what's going on here yet. The characterization and the determination of the functions of the protein components of the spliceosome is still a work in progress. So there's no way in a course like this that we can go into all those details. Just to give you an overview of how this happens and how complicated it can be. Now there can be defects in splicing. I mean, I'm saying how accurate it needs to be, but there actually are defects in splicing, and some of these defects can cause diseases. Let me just give you an example. So there are defects in splicing that can lead to disease. I'm giving you one example just to see what kind of thing can happen. But there are many that are known. The example that I'm giving you is called a thalassemia. And this is a hemoglobin deficiency. It's kind of like an inherited anemia. So if we look at beta globin, the 5' cap, exon 1, exon 2, exon 3, the poly A tail, goes through its proper splicing, and then the 3 exons are joined together, and you have the proper message to make beta globin, which will contribute to hemoglobin. So there's two different kinds of mutations and defects I want to show you. On the left-hand side, there's one example. So here we have a mutation that destroys a splice site. So in this particular example, we're looking at the intron and exon 2 boundary. So the conserved sequence is CAG, and then that line represents the exon-intron boundary, and then there's a G in the exon. So that's the normal sequence. Now, if there happened to be a mutation that occurred, and it changed the sequence from CAG to CAU, in other words, it introduced the U instead of the G, then that sequence can no longer be recognized by the small nuclear RNA, and so now it just looks like any other RNA sequence, and so the spliceosome does not recognize that boundary. And so it skips it, and the next 3', the next splice site is the boundary between the intron and exon 3. And so what happens is the splicing starts, and it skips over. It thinks that exon 2 is part of an intron, and so it skips over exon 2, and it joins together exon 1 and exon 3. And that's what's shown below, where you have now a shortened messenger RNA with only 2 exons, and of course that is not going to make a proper protein. Now there's another example where you can actually create a splice site, also by mutation. So here you have the 5' cap and the 3 exons, and this is just for illustration purposes, and what I'm showing is that in the intron there's a sequence. I hope you can't really read that. Maybe if you enlarge it you could. It would be CAUG. CAUG. Just a random sequence in the intron, CAUG. But then there's a mutation that occurs, random mutation, and what happens then is that U is converted to a G. And now all of a sudden, that looks like a bona fide splice site. So the spliceosome recognizes that, and then uses that as the splice site and not the boundary between the exon 3 and the intron. And so this splicing occurs and leaves part of the intron in. So you see the product at the bottom. You get proper splicing between intron 1 and exon 1 and 2, and then you get a defective splicing that leaves some intron in the sequence, and exon 3 is stuck at the end there, and that, of course, is not going to give a proper protein. Now, all of these, I'd say the vast majority of these splicing defects are genetically inherited. That means that the mutation is in the germline and is in the DNA. And so that means that every single RNA that's made from that gene will undergo defective splicing. And that's why you end up getting that phenotype, showing that disease. Because every messenger RNA, every primary transcript that's transcribed from that gene will have that defect and undergo the incorrect splicing. My guess is that defects in splicing take place quite often. Nothing is 100% perfect, and if we're talking about a transcript where you have to remove 25 introns, I mean, chances are there are going to be some errors at some point. But if it only happens 1% of the time, then you're not going to see the effect as a disease. So if you're making globin, and if you have a normal globin gene, you're making globin just fine, and 2 or 3 or 4% there's an error, so you still have 97, 98% of the transcripts are fine. So you can lose a little bit and still obviously not have any kind of great defect. It's when it's inherited and every single transcript is defective, that's when you see the problem. Okay, so that's defects in splicing. Now I want to go on to another property of splicing, and that is what's called alternative splicing. Alternative splicing. That is where not all exons are used under all conditions. So alternative splicing. So this is showing DNA here up at the top. It's showing the exon 1, exon 2, exon 3, exon 4, different colors. Exon 1 red, exon 2 brown, purple, and then green, and the introns which are removed are in yellow. And so ultimately, after transcription, you get the primary transcript where you have everything linked up. That's called pre-mRNA or primary transcript. And what can happen is, in different cells, under different conditions, you can get alternative splicing, which means that the exons can be used in different assortments. And this is illustrated at the bottom. So it's showing 4 alternative messenger RNAs. So on the one on the left, all the exons are included. This will make a protein. In the one next to it, exon 3 is not present. So you have exon 1, 2, and 4 joined. In the third example, you have exon 2 missing, exon 1, 3, and 4 joined. And in the last example, you have exon 2 and 3 missing. So you have exon 1 and 4 joined. Each one of these will make a different protein. And we see this quite often. 60% of all genes undergo alternative splicing, at least 3 to 4 different products, some much more. And so you can get these alternative messenger RNAs and alternative products. Each one will make a different protein. So you get 4 different proteins out of one gene. And so you get this in different cell types. So the same gene could be expressed in different cell types, but get different proteins depending on how the message is spliced. Or under different environmental conditions, you can get signals that cause the exons to be joined in different configurations. So you get multiple protein products from a single gene. It's extremely common. Here's an example of something even more complicated. I'm showing here an example of a gene called RAP53, which is related to the gene we study. But what this is showing is that there's many alternatively spliced products. In this case, there's 17 different alternatively spliced products that could be made from the same gene. So 17 different proteins. And you see these different spliced products in different cell types. So the gene is transcribed, but the proteins that come out are different. Or in response to some environmental signal, you can get changes in the patterns of the products. Now we just looked at, when we looked at the spliceosome earlier, and we saw how splicing, kind of the simple version of how splicing can take place. You know, one major question, and there's a lot of research going into this, is how these different exons are recognized under different conditions or in different cell types. So consequently, it has to be that the spliceosomes are a little bit different, or the small nuclear RNAs may be a little bit different. But the proteins that come together to make these spliceosomes have to react to the different cellular conditions in order to splice properly and get these alternative products out. So there's a whole nother level of complication that is not even begun to be understood. I want to show you one extremely dramatic example, which is really mind-blowing. So here is probably the most amazing thing that I've ever seen, or many people have ever seen, from a single gene. This is in Drosophila, but it doesn't matter. It codes receptors on the cell surface of axons. But from a single gene, a single primary transcript, they've identified 38,000 different spliced products. 38,000 from a single gene. And if you read the little abstract of the summary, there's 95 variable exons. And within those exons, there's even sub-exons. It's mind-blowing. But the fact of the matter is, from a single gene, under different conditions, 38,000 different spliced products and different proteins. Now, this is one pretty clear explanation as to why in human cells, for example, we have 30,000 genes, but we don't have 30,000 proteins. We have maybe 500,000 proteins, maybe over 1 million proteins. And so this is clearly due to alternative splicing and the ability to generate many proteins, sometimes as many as 38,000 from a single gene. So alternative splicing, extremely complicated, extremely interesting. So that's kind of what I wanted to say about splicing. We see how complicated it is. We see how, in a sense, wasteful it might be because of all the intron material that needs to be removed and destroyed. So what could be the advantage of splicing? Which I said sometimes appears to be very expensive. It is very expensive because the nucleotides are used, and all the energy and all of the polymerases that need to be used to synthesize these transcripts. So it seems to be very expensive and wasteful. So what are the benefits? So we think we understand two benefits. One benefit for sure is alternative splicing. So that alone is probably a pretty darn good benefit of splicing, that you can get so many different alternative products from the same transcript. But there's another one which is a little bit more subtle. So what are the benefits of splicing? Okay, so again, increased variability of the genome by alternative splicing. But the second one is a like I said, a little bit more subtle, but I find very interesting. And what I have here, what I wrote is it allows the expansion of functions. So if you look at the coding sequence of exons, they can code small portions of a protein. And as it turns out, they often encode some functional units or protein domains. We talked about protein domains earlier. And what we saw was that the protein domains were structural and functional independent units. That you can remove a particular domain from a protein and it will still function. So it turns out that the exons, quite often the exons encode an entire domain. So I have shown below, it's illustration, so one exon can encode a helix loop helix DNA binding domain. One can encode an alpha helix, another can encode a zinc finger, or may encode an active active site, or a motor domain of a protein. And what I told you earlier when we talked about the domains and that the protein domains can be put onto other proteins in order to allow them to have new functions. Like if you put a DNA binding domain on some protein, now it becomes a DNA binding protein. But that's not the way things happen. Protein domains don't move. What happens is the DNA moves. The DNA undergoes recombination and movement, and so you can easily get a particular exon to move into the DNA of another region of a gene and then be used in order to code a particular domain. So if you look at lots of enzymes, for example, proteases, many proteases, if you look at the active site, they're different proteases, but they have the same active site, the same sequence. And the exon sequences are the same. Or I mentioned when we talked about that, the helix loop helix DNA binding domain. They're identical between these different proteins. So they're encoded by different exons. So the exons encode these things. So what I said at the bottom is exons could provide pre-existing modules for new functions. So you don't need to re-evolve a motor domain, because the sequence is already there in an exon, the whole domain. Move it onto another gene, and now you have a motor domain on that protein. Okay, so those are two pretty clear benefits of splicing. Increasing the variability, alternative splicing, and then the use of these coding regions that encode an entire domain. Now, what about introns? Introns used to be thought of as junk DNA. So is there a function to introns? So the old idea was junk DNA, and now the more we learn, there's no such thing as junk DNA. The DNA, I mean, there may be some repetitive junk DNA that doesn't play much of a role, but for the most part, the DNA in the genome is not junk. So new ideas and things that we found is that the introns contain all kinds of things. They contain regulatory elements like enhancers. Sometimes they encode other genes. I'll show you below what we found. Some encode small RNAs, microRNAs, regulatory RNAs, lots going on. So the example I'm showing you below is P53, and these are some of the discoveries that we've made. So this shows the DNA. There's the Tata box. There's the first exon at the start site, and then there's a really long first intron, 10,000 base pair first intron, which is the size of a gene in its own. Then you got exon 2, exon 3, exon 4, and so on. But we were looking at the sequences in the intron, and what we found were 3 different elements that play a role in some function. So first of all, we found a transcriptional enhancer in the intron that's important for expression of P53. We found a gene called RAP53, which is just below exon 1, you can see that. But the promoter, it's running in the opposite direction. So you see the arrow is pointing the opposite direction, so it's transcribed off of the other strand. But still, the promoter for that whole thing is in the intron, and some of the gene is within the intron. Upstream of the gene, in fact, there's another, upstream of P53, there's another gene that's encoded. You can see by the upside down arrow, that's called WDR79. And then within the intron itself, we identified a completely novel gene with its own promoter, its own start site, and that gene encodes a long non-coding RNA, which we are now working on because we think it has some very important activity in the genesis or the induction of certain kinds of human leukemias. So we're looking at that. So introns, never say they're junk. No such thing. Okay, so we've looked at the generation of mature message. We've seen transcription. We saw the addition of the cap structure. We saw the addition poly A tail, and now we've seen the splicing. All that, everything that's happened so far is in the nucleus. Everything we've talked about has been in the nucleus. We looked at DNA. We looked at DNA replication. We looked at the telomeres, centromeres, transcription. All that's in the nucleus. The capping, the splicing, the polyadenylation, all takes place in the nucleus. So now it's time to leave the nucleus. Enough is enough. And so this is now a messenger RNA with a 5' cap, polyadenylated, that needs to be exported into the cytoplasm, where it then will interact with the ribosome and then translate protein. So in order to get out of the nucleus, it has to be exported through a very specific pore or channel, which is called a nuclear pore complex. So I just want to look at that a little bit and we'll finish up. So on the left-hand side, you see we're within the nucleus, and you see a messenger RNA in blue. It's bound by a cap, 5' cap protein that binds. There's the poly A binding protein, which I mentioned, binds to the poly A tail. And what happens is, there's also, I guess there's an exon junction complex. I don't really know much about that, so I'm not going to ask you about that. But I guess there are proteins that remain on there. I don't know anything about those, to tell you the truth. I need to learn about those. But anyhow, you have the 5' cap binding protein, you have the poly A binding protein. These participate in the recognition of RNA molecules that need to leave. And they leave through this nuclear pore complex. Once they're out of that complex, then they are targeted for translation at the ribosome. So I want to just look for a second at nuclear pore complex, but what I will say is that the passage through the nuclear pore complex is extremely selective. Only some things can get out. Not everything gets out. It's not just an open space. It needs to go out. Some things like messenger RNAs need to go out, and other things need to go in, like proteins. So all the proteins are translated in the cytoplasm, and they need to be transported back into the nucleus, like DNA polymerase. But it's highly selective because not all proteins go into the nucleus. Only the ones that need to go there. So there's a very strict selection of what goes in and what goes out. So if we look at the nucleus for a moment, it's on the left-hand side. So the nucleus is kind of cut across. The nucleus is surrounded by a double membrane. So the double membrane structure, the cell membrane is just a single, the plasma membrane is just a single membrane, but this is a double membrane. So there's an inner membrane, there's an outer membrane, there's what's called perinuclear space, which is the space between the outer and inner membrane. The inner membrane is coated by a protein, which is called nuclear lamina. It coats the inside of the nucleus along the inner face of the internuclear membrane. Turns out that the nucleus is very much more rigid than the cell membrane. The cell membrane you can easily break open with some detergents, but quite often you can You can lyse the cell, but the nuclei stays intact because it's much more supported. There's two membranes, and also the nuclear lamina serves to reinforce the structure. Also, we'll talk about this later. What's connected is the ER, endoplasmic reticulum. So the endoplasmic reticulum membrane is continuous with the outer membrane of the nucleus. But within the nucleus, or embedded in the nucleus, are what are called nuclear pore complexes. And there's thought to be anywhere from 3,000 to 4,000 of these pores in each nucleus. Now, often when you think of pore, you might think of like a little pinhole, but that's not what these things are. These are extremely, again, like everything else, extremely complex structures. Each one of these pore complexes is made up of about 150 proteins. So they're not just pores, they're actually pore complexes. And they have a very specific shape and structure, and you can see that in the EM picture on the right-hand side. These are purified nuclear pore complexes, and we're actually looking down on them. Okay, and you can see the kind of the pore in the center, and then all these structures circling it. So each one of these is an individual nuclear pore complex. So, not going to go into a lot of detail, but just want you to take a look. This is kind of the way they're drawn. So on the left-hand side, what you see is the nuclear membrane, which is a double membrane. On the bottom is the nuclear side. We know that because there's a nuclear lamina is there. And then the outer side would be the side facing the cytoplasm or the cytosol. And the complexes are many, many proteins, as we mentioned, and they have very specific structures. So on the cytoplasmic side, you have these what are called cytosolic fibrils, or filaments, which are reaching out into the cytoplasm. Then you have the core complex, which is made up of all these proteins that we saw in the previous slide, that they have a very uniform shape with subunits, and you can see that's kind of illustrated by the partitioning of this thing at the surface. And then it passes through both the inner membrane and the outer membrane, and it's held by proteins in the in the space between the two membranes. By that, you can see that little green, it's a protein complex that holds it in there. And then on the cytoplasmic side, which passes through the nuclear lamina as well, there are a bunch of nuclear pore complex proteins, and they call something that looks like a basket. And RNA molecules are allowed to get into that basket by virtue of the poly-A binding tail, or the poly-A binding protein. And if you look on the right-hand side, you have this, shows the structure. Again, just an illustration, you have the cytoplasmic filaments which stick out of the cytoplasm. You have what's called the central framework, the ring moiety, then the nuclear basket. And so this is what these things are thought to kind of look like. Now, what's not shown on there, or what's not illustrated on there, are what are called nucleoporins. The nucleoporins are those, I think those purple, meant to be those purple squiggly lines there. Nucleoporins are receptors. And they serve as receptors to be able to recognize what needs to go out, which would be a messenger RNA. So nucleoporins recognize the poly-A binding protein, for example, and then shuttle the RNA out of the nucleus. And the nucleoporins on the cytoplasmic side will recognize structures on proteins that are destined to enter the nucleus. So they're receptors that recognize specific products. So DNA polymerase needs to get in, telomerase needs to get in, histones need to get in, Now the single-strand binding protein, ligases, all those proteins need to get into the nucleus, and they all have structures that will be recognized by these nucleoporins, or these receptors on the surface. Okay, so we have made our RNA molecule, messenger RNA. We have spliced it and polyadenylated it and capped it, and we have now sent it out of the nucleus. And we are going to start looking in the cytoplasm. In particular, we're going to start looking at what happens when the RNA gets to the ribosome. Okay.